At the meeting, I think there have been three key sets of information about haemophilia B, one concerning the SPK9001 vector and two other abstracts concerning AMT060 and 061, which is AAV5 factor 9. The data on SPK9001 looks at 15 patients coming up for a year's or more treatment, and I think the important feature there is that we're continuing to see expression and that we're getting a rough idea of where they're levelling out at. And I think the figure that was given in the abstract was 23% plus or minus 10%. So I think that that's good, solid further information. The most important thing in many respects about gene therapy with AAV, the question that we've all got, is durability, stability and things like that. So any of this long-term data, although it might be a bit boring in some respect, it's actually incredibly valuable because it just adds to that body of knowledge and security about how long these treatments are actually going to last. The other important set of information that we've seen concerns the AMT060 and AMT061, so that's uniQure's program using AAV5 and factor 9. So the 60 program is a wild type factor 9, and the importance there again is about long-term data. This is data that presents patients 3.5, 4 years out and has followed them from the original trial. 2 different doses were used, but the key message is that they're continuing to express and it looks pretty stable and predictable over that period. The second bit of information is the 61 program, which is a Padua factor 9 that replaces the wild type. So essentially that's a single nucleotide difference in the whole of the expression cassette. And at 30 odd weeks into treatment, 3 patients are expressing very good levels, between 30 and 55 units per deciliter. Now, taking those two together, we can extrapolate that the solid long-term expression data that we saw in the 060 program will be reflected in the 61 program, that's the Padua variant, because there is just a single nucleotide different. So it's almost like we've got a pre-run of the Padua variant. So I think we have to take those 2 abstracts together to get the most out of it, but it looks like good data.
Well, I think with the novel therapeutics that have come to the field of hemophilia in the last few years, the bar for the expectations on outcomes with gene therapy for hemophilia has really been raised considerably. So safety and efficacy have been shown now, over the past decade. But I think what people are really interested in now is what's possible with gene transfer in hemophilia. So there's considerable focus on the predictability of the levels that patients can achieve, and then also questions about the durability, and then of course everyone's interested in what kind of level of correction patients are able to achieve. So a couple of updates at this meeting which I think are really important. So the uniQure phase 1/2 program for what was called AMT060, which was their AAV5 vector system with, at that time, a normal factor 9 transgene. So we now have seen the presentation of the long-term data, if you want to call it that, over the last 3 to 4 years from the 2 cohorts in that study. And in the highest cohort, patients were achieving expression between 5 to just over 10%. But we've now seen at this meeting that those levels are durable over that 3 to 4 year window. And most importantly, the bleed control appears to be really excellent. A very low incidence of bleeding after gene transfer, and really a remarkable reduction in the amount of factor 9 utilization. The update that's being presented today actually now comes from the AMT061 program, which is the same vector as the AMT060, but this time substituting a hyperactive factor 9 transgene, the so-called factor 9 Padua. So it's the same vector system, just new transgene, and the 1-year data from that phase 2b study is showing that these gentlemen are expressing in the range between about 35% and into the normal range, above 50%, with levels that have been stable over the past year. So I think that shows I think the ability to move patients into the normal range. And secondly, I think we can draw on the expectation of the durability that we saw on the AMT-060 program and likely translate that into the expectations for the 061. So I think this is pretty exciting. Of course, the phase 2b patients at this level of expression, essentially no breakthrough bleeding that has clearly been attributed as true bleeds, and a remarkable reduction again in the amount of factor IX utilization.